Hello members, today our topic is going to be airpin countercurrent multiplier. Airpin countercurrent multiplier, here we are going to look at the biology occurring in the kidney, but specifically in what we call the kidney nephron. And then when we enter the kidney nephron, we are going to dwell more on a particular portion of the kidney nephron, which we are calling the loop of Henle. I want you to open your ears, even eyes properly. Just enjoy the lesson. Enjoy the biology. This biology is very, very, very important. You always move and urinate. There you are going to appreciate that portion which you are going to talk about. Otherwise, we would be urinating every time, and the body would be dehydrated. But again, we are seeing our body remains hydrated because of this biology we want to look at. So follow me up and enjoy the biology. As you are beginning our lesson now, you need to remember this. It is very, very important. Our kidneys have a remarkable mechanism for reabsorbing water from the renal fluid or from the tubular fluid. And now the way how it is reabsorbing the water back to the body, that is now our point of concern. And that mechanism is what we are going to talk about as our countercurrent multiplier or multiplication. The multiplying, you are going to see it ahead there. How the salts are going to multiply. We shall see somewhere in our diagram there where the salts in the descending limb at the top when the concentration was around 300. But by the time we are going to reach downwards at the bottom, it is going to be around 1,200. That kind of multiplication is what a biologist now is going to call multiplier. Then countercurrent, as you are moving ahead there, you will remember this. Countercurrent, we are going to look at the fluids flowing oppositely. When you look at the two sides, the ascending and the descending, the fluids are moving oppositely. You discover the fluids in the descending are moving from top, the bottom, but again when you are looking at those ones of the ascending, they are moving from bottom to top. Then that kind of arrangement is not only in this subtopic, but we have also seen it under fish when the blood is flowing opposite to the water in the gill filaments, and also seen somewhere under circulatory system as we organisms, like especially animals, we are economizing on the heat loss. We are seeing that the blood flowing downwards in the arteries are moving parallel, those ones which are taking blood back from the extremities. So we are seeing that we economize the heat from being lost. We see that the heat moves from the arteries as blood is going to extremities to the veins, making the blood going to our extreme ends to be cold and the blood coming back to be warm. Are you seeing that kind of biology of countercurrent? Even here is a biology you are going to apply, but the difference here we are going to look at now the salt concentration. When you are looking at circulatory system, you are looking at the heat, how the heat in the blood, heat energy. Now again, when you are looking at counter from the fish, you are looking at concentration of oxygen. So try to be attentive as you are moving ahead. The question is going to be, how does this one occur? Let me begin with occur, then we shall come back to what we are calling the countercurrent, or you are calling the countercurrent. Now, the first question of how it is going to occur, that is, how does it occur? We are going to begin by studying this structure properly. Now, members, what I've drawn here, this one we are calling it now the loop of Henle. The loop of Henle, remember, it is U-tube. Is a U-tube like that. Loop of Henle is a U-tube, don't forget. The loop of Henle on top of that is having what we call a descending limb. is having what we are calling the descending limb, and is having what we call the ascending limb. Remember, the descending limb, as you are seeing, it is thinner. When you are drawing it, we are indicating that is what? We are indicating that is what? Is thinner. Then, when you look at this one, the ascending is thicker. And see, is thicker. Then on top of that, the structure of the loop of Henry, it is long, and on top of that, it is hairpin-like. Then you have to understand those ones. The ascending is thicker, the descending is what? Is thinner. Then on top of that, we are seeing that the renal fluid, this one is the tubular fluid now, is entering here. This arrow is showing it entering. So here we have what we call the renal fluid, or which you can even call the tubular fluid now. And here it is leaving. Now what happens in this mechanism here? Take note of that. But before we see even how it happens, let us first take a note of the following. You can see that as you're moving from top here downwards, we are seeing that the concentration of the salts is increasing. From up here, we have 300. Then when you are moving here, we have 1,020. But again, when you look at this one, as you are moving from top, we have 100 here. From top, we have 100. Then when you come here, we have 1,000. Take note of that. This implies what? That in the descending limb of Henry, the concentration of the salts are keeping on increasing from top to the bottom. But again, when you come to the ascending, the concentration of the salts ascending, you are going to discover that the concentration of those salts keeps on decreasing as you are moving from bottom to the top. It is clearly reflected in our diagram there. At the bottom we have 1,000, but as you are moving upwards, we are seeing that we end up with a concentration which is about 100. Still maintaining the difference of 200, try to compare the figures on left and right. You'll discover that there is a difference, osmotic difference created there, which is of around 200 units, which is very, very vital as we are going to look at our mechanism. The concentration of the salts in the descending limb here is opposite to the concentration of the salts in the ascending. Now, all the above mechanism is going to drive us back to how does it what? Occur. But before we continue with how it is occurring, take a note of these members. Look at the red arrows here, my red arrow here, this one. They are showing that the salts are coming from ascending to where? To the medullary region. Salts are coming from ascending to the medullary region, represented by the red arrows. But again, when you come here, we are seeing that the water is coming from the descending to the medullary region. Take a note of that. From ascending, salts are coming out, meaning the ascending is permeable to the salts. But again, when you look at descending, the water is coming out. This implies, guys, that the descending is permeable to water. And we are going to see that the water coming here is by osmosis. You can look here at our key here. We are saying the green arrow. This green arrow is showing osmosis. Then when you come back here, our red arrows of the salts, for them they are showing what we call active transport. Don't forget that. So we want to paint our image to understand it properly. This countercurrent multiplier properly. Members, we are going to begin by focusing at the ascending what? at the ascending limb. As the renal fluids are moving, now we are looking at how it is occurring. The whole mechanism is all about generating an osmotic gradient or a big concentration of the salts such that water can be reabsorbed back to the body. Remember, we have said with the essence of this mechanism, we have already told you that the mechanism is important because it prevents you from producing liters and liters of dilute urine every day. And it is the reason why we don't need to take water every time. Because we are capturing water, we are reabsorbing water back to the what? The body. And then we are saying the mechanism is all about generating an osmotic what? Gradient in the loop of Henry that can enable reabsorption of what? Of water. This implies what? That the current multiplication here, or the counter current multiplication here in the kidneys, is going to be simply a process of using energy. The energy we are going to see active transport here. The active transport here is applying what we call the sodium potassium what? Pump. So here we are going to see what we call sodium potassium pump at the ascending, which is using energy, and this expression is how we are calling it active what? Active transport. Now we are going to see that the counter current multiplication we are talking about is going to be simply the process of using energy to generate an osmotic gradient that enables you to absorb water from the tubular fluid and produce concentrated what? Urine. That one I told you at the beginning, and we are saying it is occurring where? It is occurring in the kidney nephron, at which point the loop of Henry. The question is going to be, why is this one called countercurrent? Members, countercurrent. So, members, one question was asking here, why are we calling it countercurrent flow system? The best answer is going to be here. It is countercurrent flow system because we are seeing that the fluids are flowing oppositely. You can see that these ones are moving downwards, but again these ones are moving what? Upwards. So that opposite flow of the fluids is the reason as well we are calling it countercurrent. You will discover that in all countercurrent systems there is flow in opposite what? There is flow in opposite directions. Now take note of that flow of the opposite what? Opposite direction. This explains why in most cases you'll find most biologists will tell you it is countercurrent because the fluids flow in opposite what? Direction. We are saying in the ascending the fluids are flowing upwards. But again when you look at the descending, the fluids are flowing what? Downwards. Let us try to look at this point here. What are you seeing? You are seeing that here we are having 100 at the top, then downwards we are having 1000. This implies that as the concentration is increasing in the descending, the concentration again members, as the concentration increases in the descending from top to bottom, the concentration and decreases from bottom you have less salts in the ascending, you have more salts in the what? In the descending. So members, our idea was to clearly show how does the mechanism occur. Now our mechanism here, we are going to agree that it is going to begin with now the ascending. We are saying the ascending limb here, this ascending limb, it is permeable. It is permeable to the salts. And being permeable to salts is having what we call the carrier proteins. Is having what we call the carrier, is having carrier proteins. Carrier proteins. And these proteins work as ionic what? Ionic channels. Working as ionic channels, meaning they can carry the ions. Which ions Our first ion here, we have what we call the sodium ion, and another one we have what we call the chloride ion, we have what we call the potassium ions. We are seeing them here moving out. Now, because of the presence of the protein channels here and the permeability of the ascending limb, the salts are actively reabsorbed, or the salts actively move away from the ascending to the neighboring medulla region here, neighboring medulla region. Now this implies what, members? That as the fluids are moving from this side of the what? Of the descending, but remember they are going to move like that. They are going to move like that, like that. Those are fluids moving. Which fluids moving have been carrying the what? The salts. They have been carrying the salts. Now when they reach here, this membrane here is permeable. The membrane here is permeable. Why is it permeable? Because of the protein channels. So active transport occurs here, and the salts are going to move out like that. As they are moving out, the sodium ions, the potassium ions, the chloride what? Ions. You can see chloride ions, potassium, and so on. They are coming out. When they come out, they are going to concentrate in this region of the medulla. They are going to concentrate in the medulla region here. As they are concentrating in the medulla region, what are we doing? We are increasing the concentration of the salts in the medulla region. When you increase the concentration of the salts in the medulla region, remember we are saying this one of the descending is permeable to water and permeable to few ions. So the water is going to move out of the descending limb by osmosis. So the oral mechanism is all about concentrating in the medulla region with a lot of salts. How are you concentrating them with a lot of salts? Salts are actively moving from the ascending via the carrier proteins. The salts are sodium ions, potassium ions, and other. They come out by active transport. It is a sodium potassium pump which is pumping them out, and then they go to the medulla region or the interstitial space. Now when they come here, you see the air concentration is increasing. When the air concentration increases, now we are going to see that water is going to be osmotically moved from the descending to the medulla region. So the instance of this one, we are concentrating salts here. You are forcing the water to move by osmosis from the descending what? From the descending limb to the medulla what? The medulla region. Now as the water is moving out, as our water is moving out, that is water moving out, what are we seeing? The concentration of the salts in the descending limb is going to increase. A question is going to be, when you look at the medulla concentration and descending limb concentration, almost the concentrations are what? The same. We have 300 here for medulla region, but again even here you have 300. Then you have 1,000, 1,200 here, and even here you have 1,400. Why is that the water doesn't move backwards? The answer is going to be, the best answer is going to be, this one is having salts like these ones, but this one is having a lot of water. Remember, these fluids are going to form the urine in the collecting duct. So as the water is going there, some water is reabsorbed back to the body. As it is being reabsorbed back to the body, what are you doing? You are increasing concentrations of the salts in the descending what? The descending limb. Now, this implies that the more the fluids are coming towards ascending, the more sodium and potassium ions are going to be removed, and the more concentration in the medulla region will become. Then the more water will be removed from the descending limb. So the increase in the concentration of the salts in the descending limb is as a result of the water moving osmotically out of the descending, being permeable to water to the medulla region, in causing increase in the concentration of the salt. You can see why are we seeing increase the salt concentration here. This is because of the osmotic withdrawal of water from the descending. As water is moved from regional of low concentration of the salts, regional high concentration of the what? Of the salts. Then here the ions are moving against their concentration gradient. Now they can ask you, how does it occur? How does it occur? This question here, how does it occur? Now, how does it occur? Our main emphasis will be on one, the ascending limb, being what? Being permeable. Being permeable to salts. Because of the ascending limb being permeable to salts, what happens next? The sodium ions, potassium ions, and chloride ions move from the ascending limb to medulla. What happens there? Concentration of the salts increases. When the concentration of the salts increases, what happens? Water is osmotically withdrawn from the descending limb of Henry now. Now you are seeing that the whole idea is all about this one being permeable to the salts, and the salts are. So how it is occurring, members, I think you have verified that. You are going to talk about for the sound as describe, explain the counter-current multiplier. Your focus will be first on the permeability here over the ascending limb, that this one is permeable to the sodium, potassium, and chloride ions. Why permeable? Dissolving what we call the sodium-potassium pump via what we call the channel proteins. Then the ions are going to move from the renal fluid, the tubular fluid of the ascending limb to the medullary region. When now they reach the medullary region, the concentration of the salts in the medullary region is going to increase. When the concentration of the salts in the medullary region increases, you'll find that water is going to be withdrawn from the descending limb of Henry by osmosis. So from water from the descending, this one is mainly by osmosis. Then the salts from ascending, this is mainly by active transport. Active transport is energized by energy. And then here, where it is occurring passively, no energy is needed. Members, when you look at the oral diagram here of the countercurrent flow system, you'll discover that there is a difference of 200. When you come to the descending, you have 600 here. Then when you come to the ascending, you have 400. When you come to descending back here, we have 900. Then even here, you have 700. Here you have 1,200. Here you have 1,000. This means there is an osmotic gradient which has been maintained in the whole process of 200. So we have our 200 there being what? Being maintained. 200 units. So they are being maintained in the whole system. How are you maintaining? You are losing water in the descending, causing increase the concentration of the salts. Which salts are going now to come in the fluids here. When it comes in the fluids here, again is reabsorbed back via the ascending limb.
Then another thing to note is that more water is withdrawn from the descending.
More water. Then here more salts. So more salts is being reabsorbed, more water is being reabsorbed. Reabsorbed by osmosis. This is by active transport at the ascending. So the more water is removed from the descending limb from top to the bottom, as its concentration increases from top to what? To bottom. Don't forget that we have 100 here, so we have 300 here, and we have 1,200. Then from there, another thing to note, as you are moving, some ions can even move out of the descending limb, but water is going to move much more rapidly from that limb of Henle. So we will discover that the water does not dilute the medulla because it is taken away by the blood capillaries to the blood. How comes if water is coming out doesn't dilute this one? Or the water, remember this one, is going to be surrounded by some capillaries. You will discover this one is surrounded by capillaries like that. Don't forget that in biology, we have some capillaries surrounding like that. Many capillaries, we call them the vasorecta. These capillaries here, these capillaries are connecting the main blood stream. When the water comes here, the numerous blood capillaries which you are showing here, they will just carry the water to the main blood stream or the circulatory system. So the water, however much the water comes out, the water doesn't dilute this salt concentration. This one is maintained thoroughly. That question can be asked to you. Why does the water, if it's coming from here, doesn't dilute the whole thing? Our argument will be the water is taken by the vasorecta or the capillaries. Now let us also look at our keywords here. The first keyword they are talking about hairpin. Hairpin, that one we are referring to Henle, this loop of Henle, being long at the same time being hairpin shaped. That is a hairpin. Actually, hairpin is arising from the structure and appearance of the loop of Henle. Loop of Henle, this is the one we are talking about. The loop of Henle is a part of the nephron. Countercurrent, that one we are referring to the fluids flowing oppositely or generating energy that can create an osmotic gradient. Multiplier, we have seen at the salt concentration increasing. That is the 300 then to 1,200. You are multiplying. Then medulla, this is the medulla region. All of this region surrounding the loop of Henle. Here, that is the medulla region. Then when we talk about interstitial space, medulla region, that one, all of it, this space here, that is in interstitial space. Tubular fluid, that is the fluid flowing. The one shown by the arrows, flowing in the what? In the loop of Henle and the overall nephron. Active transport, we are seeing it at the ascending. The ions, that is the salts, sodium, potassium, and chloride, are moving via the carrier proteins, which form what we call the sodium potassium pump, which require energy. So that is active transport, moving against concentration gradient, but by using some energy. Of course, you are moving from low concentration to high concentration, so your movement must be helped by some energy or fueled by some energy. Sodium potassium pump is occurring here. That is one which is doing active transport. Then we have what we call the protein channels. Those ones are the ones which are carrying the sodium, potassium, and other salts around the what? Around the ascending limb. Then hyperosmotic, this one means high concentration. So the medulla becomes hyperosmotic here to allow the water to move by what? By osmosis. That one is hyperosmotic, highly concentrated here. Hyper means high. Osmotic, of course, you know the difference in concentration. Osmotic gradient, that is mainly the difference in the concentration. Urine, that is urine now coming out with less water. Reabsorption, salts coming out, that is reabsorption. Water coming out, reabsorption. Why already these materials are being reabsorbed for the second time. The first absorption was already in the, that is in the small what? Small intestines. Small intestines. That was the first what? First reabsorption. Then here we are reabsorbing them for the second time.